All right, so welcome to the series of lectures on the liver. This is going to be a long set of lectures, and I probably won't be able to get through all of this in 1 week. We have to cover the liver and the gallbladder, both physiology and anatomy, and then look at some common hepatobiliary pathologies. So I want to kind of just go over the liver structure, the whole structure of the biliary tree, the functional unit of the liver. This is called the liver lobule. So we'll look at that, and then the classic liver cells or hepatocytes. We'll look at their physiology. Look at the major functions of the liver, and there's a lot, and that involves glucose, protein, and lipid metabolism, as well as detox and bile salt formation, and many, many other things. So we'll have to just sort of do a survey of that, and then look at some of these common liver disorders, like the different types of hepatitis and fatty liver and gallstones and so forth. So there's a lot here. The liver, this essentially, in English, the liver to live, this is the provider of our life activities. So we've been looking at the digestive system, and remember that all of those nutrients that were absorbed through the portal system, so from the intestines and from the stomach and whatnot, they go into the portal system and they immediately go to the liver, with the exception of large chain fatty acids, which go through the lymph directly back to the circulatory system via the thoracic duct. But with glucose and your vitamins and all your proteins and so on, and short chain fatty acids and so forth, all of this goes to the liver. So the liver is really the processing center for everything, and then that's going to go into systemic circulation. I just put a couple of little interesting things here. Historically, people have understood physiology on very, very subtle and deep levels, and in particular in ancient Greece, there was this myth of Prometheus, which is really an image of the whole liver process. I will come back to this because right now it might be a little premature to look at the meaning of this, but this kind of will give us a context of the liver in the broader sense as we go. The liver is the second largest structure, largest organ in the body. The skin's the largest, and it's also the largest gland. So notice that it occupies a tremendous amount of space here in the upper abdominal cavity. So the liver and the stomach sort of form a pair this way. It's asymmetrical, so again, this is different than the brain, the central nervous system, and the kidneys. The average weight's about 1,500 grams in the average adult male, a little bit smaller in women. There are a couple of surfaces. So the upper surface is called the diaphragmatic surface because the diaphragm rests right on top of the liver here, so it kind of wraps around and rests on top. And the liver shape is really determined by the diaphragm. So as the diaphragm contracts for breathing and contracts downward, it actually squeezes the liver and has sort of a pumping action. So breathing has a relationship here to liver function. As the diaphragm relaxes, the blood, portal blood, and everything else is sort of sucked into the liver. And so this allows sort of a circulation of fluids and whatnot throughout the whole abdominal cavity. The lower surface is flat, and it's really determined by the organs pressing against it. So we have the gallbladder over here. Notice that the colon, of course, wraps around this way, the transverse colon. Here we have the ascending colon and then descending colon, and stomach on this side, and so forth. So basically, what you kind of get as an image here of the liver is it's very fluid and plastic, and it's sort of shaped by the other organs around it. So it shows you this capacity of the liver just to have a strong association with water, and we'll have to come back to that and explore that in detail. A couple of important landmarks on the liver. So first of all, the liver is divided into two lobes, the right and the left lobe. And again, this is patient's right and patient's left. And the gallbladder is down here. And then there is the visceral peritoneum, covers all the surface except for the upper portion here of the diaphragmatic surface. This is called the bare area. There's also a fibrous capsule that wraps around the whole liver. There are ligaments which are used as landmarks. They're not true ligaments. They're actually remnants of different embryologic structures. So the falciform ligament, for example, divides the right and the left lobe. The round ligament here, and then a little coronary ligament. if you look on the underside of the liver, you find that the right lobe is actually further divided into two lobes. So the so-called caudate lobe and the quadrate lobe. And what you see right here is the where all of the different vessels enter the liver, like the portal vein and so on and so forth, right here between the two lobes. Okay, so here's a little bit more on the ligaments. The round ligament, for example, is the remnant of the umbilical vein of the fetus, and so on and so forth. So you can read through that. These are more just like identifying landmarks. The blood supply is fairly simple. So there's basically, first of all, let's start here off the aorta. The hepatic artery branches off the celiac trunk, and that extends and gives fresh arterial blood to the liver. Sometimes there are branches of the superior mesenteric artery, which supplies the upper intestine, also supplies some blood to the liver, but usually it's just the hepatic artery. The portal vein, this is coming from all your intestines, is bringing essentially deoxygenated blood, venous blood, but it's loaded with all these nutrients from the digestive tract. So the hepatic artery gives you the fresh oxygenated blood, and then the portal vein, the venous blood. About 60 to 80% of the blood flow to the liver is from the portal vein, and only about 25% is from the hepatic artery. So you see this interesting phenomenon where the liver is very good at living in a state of relative hypoxia. So unlike organs like the kidney, where we'll see there's an enormous oxygen uptake. So kidneys use up about 20% or so of your cardiac output and oxygen supply. So, but the liver uses up much less here. The hepatic veins then will look at the inner structure, but they're going to collect all the blood as it's filtered through the liver and deliver it back to the inferior vena cava. So that's the venous drainage of the liver. And I think we have a different picture down here. So you can see the portal vein coming in, the hepatic artery coming in. It branches here into right and left hepatic arteries, and then the hepatic veins draining into the vena cava. There is a parallel network of little bile ducts in the liver, and they're all collecting around, and they're going to end up flowing into what's called the right and left hepatic ducts, which then merge into a common hepatic duct, and then this is going to go down as a little cystic duct onto the gallbladder, and this will be the common bile duct that will lead the bile through the pancreas into the intestines. So interestingly, although we often talk about gallbladder as a separate organ here, this is sort of a storage sac for bile. Notice it's more of an overflow sac. For the actual bile is made up in the liver, and it's all these little canals that collect the bile and lead it down. So a lot of people, when they have gallbladder problems, and that would be, for example, gallstones, where the bile becomes sluggish, the problem really involves some degree of cholestasis up here in the liver. So really this sac, anatomical sac, is not the true gallbladder in the sense of traditional medicines like Chinese medicine. They're talking about a process that's beginning up here in the liver, but also beyond the liver with bilirubin and so on and so forth. So I'll go more into that later. The area where the portal vein, hepatic artery enter and the bile ducts leave, this is called the hilum of the liver. And these essentially divide, all of these divide into left and right branches, as we saw, supplying the right and left lobes. The lobes can be further subdivided into 8 smaller functional lobes, and these are called hepatic segments. And each segment is supplied by a secondary or tertiary branch of the portal, what's called the portal triad. That would be the hepatic artery, the portal vein, and the bile duct. So each one of these segments really has its own little supply of blood and bile that leaves it. And this is important for surgical procedures, because if you have a tumor or something in the area of the liver that needs to be resected, you can take out one of these segments and leave the others intact. And as we'll see with the liver, there's a tremendous regenerative capacity, so you can take out 2/3 of these segments and they will regrow. So you can have regrowth of them as long as there's no scar tissue preventing that. And that's why in cirrhotic livers, with liver cirrhosis, there's scar tissue, and that really prevents the regrowth of liver tissue. The liver is a major lymphatic organ. About 20 to 50% of the lymph received by the thoracic duct comes from the liver. So it's not shown here in this picture, but the liver really stands here as a major lymph-producing organ, and it's contributing all of this lymph to the thoracic duct, which is then going to go into the subclavian vein. There are superficial lymphatics located on the outside of the liver, and then deep lymphatics, and these accompany the hepatic veins. Most of the lymph is formed in this little inner space. I'll talk about this later. That's called the perisinusoidal spaces of Dis. These basically is where the lymph is produced. It's secreted into these little spaces, and then it drains out to the deep lymphatics. There are hepatic lymph nodes found in the lesser omentum. This is important for a number of different reasons. One is, remember that adenocarcinomas in particular like to metastasize through the lymph. So colon adenocarcinomas, for example, will metastasize through the lymph. And unfortunately, they will make their way into the liver. And so the liver is a common site where these metastases will end up. So we'll see that although the liver can develop cancer on its own, the majority of liver cancers come from metastatic cancers. Now, the bile flow in the gallbladder, bile is produced in the liver, and they collect in these little ducts called the bile canuliculi, and I'll show you pictures of those coming up. And they merge to form the bile ducts. So I think the best picture might be this one down here. So the bile ducts all collect into a right and left hepatic duct. These are from the liver. These merge to form a common hepatic duct. And then branching off of that is a cystic duct, which leads into the gallbladder itself. And then coming down off the common hepatic duct would be a common bile duct. And that's going to go into the pancreas and merge with the pancreatic duct. This is where the digestive juice and enzymes comes out. And then they both merge onto the hepatopancreatic duct, or ampulla, in the sphincter of Oddi here. And so the pancreatic juices and bile will be essentially secreted at the same time. And so they will come out, and the signal for this, remember, would be, for example, fat going into the intestines and releasing neurohormones like CCK, cholecystokinin, and so forth. Notice that stones, gallstones, they often form in the situation where, and we'll talk about this later, where there's often the bile becomes, most of these stones are actually formed from cholesterol, and the cholesterol begins to precipitate out of the bile. So normally there's cholesterol in the bile, and it's kept in solution, but if there's a little bit not enough phosphorus, the phosphorus will allow the cholesterol to precipitate. And then you'll start forming stones. And they can form anywhere along the bile ducts, but in particular, stones will like to collect in the gallbladder. This is an area where the bile will sit, often for many hours. And so it'll sit in here, and unfortunately then, when you eat a fatty meal, the smooth muscle around the gallbladder sac, and it'll contract it, and then these little stones can come out and start lodging in the bile ducts. And maybe down here, even blocking the pancreatic duct. This would be quite severe because this could back up and cause acute pancreatitis. So this is where we're careful about patients with stones, and the way we diagnose this is with ultrasound. So if we see stones on ultrasound, we need to look at their size. Sometimes it's just gravel, little tiny ones, and those are usually okay. They'll pass through all right. But if you do therapies that stimulate the gallbladder and the person has larger stones that have been kind of sitting there for a while, they can potentially block the bile ducts. And this can cause severe inflammation, pain. Bile will start to back up into the liver as well as the gallbladder. Infection could arise, and then this could burst. And we have a situation called acute cholecystitis. So I'll review all that when we get to the gallbladder. So that's a basic overview of the bile ducts. Okay, the liver has nerves, like we've seen with all the other organs, both sympathetic and parasympathetic. We'd say that most of the major nerve supply is parasympathetic, and that includes innervation of the gallbladder and the bile ducts. So the smooth muscle there is stimulated by the parasympathetics. Sympathetics will regulate the flow of arterial blood into the liver. So if they're activated, the sympathetics will contract, the vessels, less blood flow. If it's relaxed, there'll be more blood flow. Interesting research on rats showed that every function of the liver is under the control of the autonomic nervous system and with connections with the hypothalamus. So the autonomics really help to regulate all of this, and we've seen that tremendous autonomic presence in the digestive tract and the importance there as well. Now, morphologically, there is a basic structural or functional unit of the liver made of many, many different types of cells, as well as blood vessels, nerves, and lymph vessels. And it has a classic hexagonal or polyhedral shape. And this is called the liver lobule. So let's look at one of these liver lobules. Here's one right here. So all these little dots are actually cell nuclei, so these are all cells. So these are different types of cells that we have in here. At the corner of the liver lobule, so they're kind of three-dimensional, of course, they're kind of like columns, but at each corner there's what's known as a portal tract or portal triad. And that consists of a branch of the hepatic artery, that's in red here, that's supplying fresh arterial blood to everything around it. A branch of the portal vein, remember this is bringing in the nutrients from digestion, and then a bile duct, which is going to go down and these are going to collect into the left and right hepatic ducts, the common hepatic duct like we just reviewed, and so forth. So that's a portal triad. So portal triads are these three vessels at the corners of the polyhedra of a liver lobule. And in the middle there's the central vein, and this is going to take all the blood out and move it into the vena cava. Okay, so the way it kind of works here is that let's follow blood, for example, from the intestines coming in the portal vein. So it's flowing in here. It's going to flow down these little tracts, and this is lined in endothelium towards the central vein. But on the way, it's going to encounter these cells right here are the classic liver cells called liver hepatocytes. It's going to encounter the hepatocytes. And the hepatocytes will take nutrients out and put things in to this little blood flow. So this is essentially like a giant filter. So it's not actually filtering through the cells, although the cells are actively transporting and then secreting into this flow. And then all of this, as it moves its way through, is going to collect in the central vein and then leave the liver. So hepatocytes are doing a tremendous amount of work here on what's coming in from the portal vein. So taking up glucose, for example, taking up proteins or amino acids at this point, and nutrients, and then putting proteins in the blood, maybe adding glucose to this little stream, and then it enters the central vein and exits the liver. There are other cells we'll look at here like Kupffer cells, and these are actually immune cells. They're macrophages that are filtering and essentially eating up any antigens that come in. Interestingly, the portal vein has a fairly high amount of bacteria that comes in from your gut, and the Kupffer cells filter all of that out. This is one big problem with liver failure, is that amongst many other things, the bacteria from the gut will come in and not get filtered because the Kupffer cells are not functioning in liver failure, and then you're going to get bacteremia and maybe septicemia in the blood from that. The hepatic artery is providing fresh arterial blood to the hepatocytes, so it's kind of going on the backside here. And then you'll see there's little specialized cells inside here, and these specialized cells are part of the bile secreting system, and they're all going to collect these little bile ducts and then lead out of the liver lobule. So very interesting phenomena that you see is there's two directions of flow here. One is from the portal vein towards the central vein, and this is going to be part of what we might call the anabolic liver process, where everything built up in the hepatocytes is added to this and it goes into the central bloodstream. The other flow is opposite through the bile canaliculi, and it goes this way. So it's kind of going out and then going to go down. So this is what we might call the inward pointing arrow, the more centripetal arrow here. We can call that the true liver process. And this other arrow of activity that goes this way we can think about as the true gallbladder process. These layers of hepatocytes are called hepatic plates. So they look like little plates because they all line up this way. The sinusoids are between the plates. So that's these little regions here. Those are sinusoids. And the sinusoids are really lined by endothelium. So these are blood vessels. Okay, and then the bile canaliculi have their own cells called cholangiocytes that line them. So here's another sort of functional diagram. Here we have the liver lobules, classic portal triads at each corner. Here we go, another portal triad here. So hepatic vein is dumping its blood into the sinusoids, making its way towards the central vein, and the hepatocytes are taking things out and putting things into that flow. Hepatic artery is giving fresh blood to the whole process. And then the bile canaliculi are going the opposite way. So they're going to flow outward and out of the liver process. So really two activities. One, inward concentrating, true liver, and the other outward, more removing bile and whatnot from the liver. That's true gallbladder. And I mention this because herbally, the treatments are quite different. The herbs that we use to treat the liver process are going to differ significantly from the ones treating the gallbladder process. And this often gets confused because people often say both are liver herbs. But interestingly, by action, there's two different sets of herbs that work in the liver. So this just summarizes what I just talked about. Okay, so I don't need to review that. All right. So here's just other pictures. This always helps to see these big pictures. So here we have the two lobes of the liver, and we've taken a big chunk of that out, and we see if we look at this more closely under the microscope, you see these lobules. And the lobules each have a central vein in the middle and the portal triads at the corners. And the blood is essentially flowing from the portal system portal system into the central veins. Okay, so here we have that classic draining this way. There is a concept here that will be important for pathology, and that's the idea of what's called the portal asonis. So the portal asonis is really the tissue supplied by the terminal branches of the hepatic artery and the portal vein. So here we have the hepatic artery. These tissues are going to get the highest amount of oxygen, and as we go towards the central vein, less and less oxygen. So although I said the liver prefers hypoxia, it's possible that areas of low oxygen will be damaged first before areas of high oxygen. So these are the cells that will receive the nutrients first, and then these will receive it last. So this will be important when we look at some pathological conditions. Okay, and here's just a nice blow-up of that whole process. Notice the bile canaliculi between the hepatocytes here. And hepatocytes are very interesting cells because they are loaded with all sorts of enzymes and metabolic machinery. Pretty much every biochemical process your body does can be done in the hepatocyte. So we have a major metabolic machinery here. Very different than, like I've compared many times to a neuron, which has a very specialized set of enzymes and activities. Okay, I think that's pretty clear here. Notice I mentioned that space of Dis earlier. So here's a sinusoid. So here's the blue, the sinusoids, and they're lined in endothelium. Here's a Kupffer cell, a macrophage. And then notice there's a little space between the endothelium and the hepatocyte. And this is the space of Dis. This is where the lymph collects. And this will actually drain itself out into a different system through the lymph nodes. So that's very important here. This space also, there's also little microvilli on the surfaces of the hepatocytes. And occasionally you'll see other cells in here. In fact, one cell called a stellate cell, or also known as an ito cell. And these can store fat. They also can become activated in inflammation and can create scarring, unfortunately, and lay down scar tissue. So those would be cells found in the space of Dis periodically. And they're mentioning here also reticular fibers, which are protein strands helping to hold the structure, the cellular framework of all of this together. Hepatocytes, like I've mentioned here, perform the major functions of the liver. They comprise about 60% of the liver cells and about 90% of the liver's volume. And they're very rich in metabolic organs like mitochondria and Golgi apparatus, like I mentioned. And they have a lot of glycogen storage as well. So they store up glycogen and then occasional fat droplets. Now the glycogen storage, we'll look at that in more detail. It's interesting because it has a 24-hour rhythm. So basically, glycogen storage is going to, glycogen, which is a storage form of glucose, kind of like starch, will begin to decrease from 3 a.m. to 3 p.m. And then from 3 p.m. to 3 a.m., glycogen stores go up. So the liver, interestingly, towards the evening hours begins to swell because these cells are becoming stuffed full of glycogen. And you can see them under a microscope. You can stain it and get these little starch crystals. As you need glucose and your blood sugar falls, especially when you're active during the first parts of the day, the liver actually releases its glycogen and it shrinks a little bit. So it's a very interesting phenomenon, the liver expanding and shrinking because of the glycogen. Unfortunately for a lot of people, the liver can also store fat droplets, and this can crowd out the glycogen, so you have difficulty storing glycogen, and that's going to result in some blood sugar regulating problems. But the cells will swell as well, and that will shut down a lot of the metabolic machinery. And this is known as fatty liver disease, and this is a really big problem. I think the estimates are about 40% of the US population has some degree of fatty liver. And this will significantly impede the liver's ability to do all of its metabolic activities. So there's different surfaces, the sinusoidal surface, the lateral surface, and canalicular surfaces of the hepatocytes. So notice on the canalicular surface, this is where the bile is secreted, so bile is secreted into that. And then the sinusoidal surface is where nutrients from the endothelium, from the portal vein, for example, will come in and go through the space of Dis and then get absorbed into the liver cell. And then the hepatocyte might actually add nutrients that will go into this sinusoid. And this will all drain back to the central vein. Notice the ido or stellate cell found here in the space of Dis. And here we have a Kupffer cell. There's a macrophage doing its macrophaging stuff activity. All right. There are tight junctions between each of these, and that's represented in the green here. Tight junctions between each of these cells, which keeps them highly tightly together, so it prevents things from leaking between hepatocytes. Okay, the endothelium, I should say, the capillaries here in the sinusoids are very leaky. So this is one of the leakiest capillary networks in the body. And that allows very large molecules, proteins and whatnot, to actually pass between the sinusoidal space into the space of Dis. And there's no basement membrane either, so this is a very important concept. Kupffer cells, I mentioned, are part of the phagocytic system. They're kind of a in-place macrophage system called part of the reticular endothelial system, RES. And if a person receives a liver transplant, interesting that the liver is filled with Kupffer cells of the recipient after some period of time. So you begin to kind of reclaim the organ by putting your own Kupffer cells in there. And they release TNF-alpha and different interleukins and interferons. The ido cells, I said, are in the space of Dis. They contain fats, and they store things like vitamin A. This is where vitamin A is stored in the liver. The liver is very efficient at storing vitamin A, as well as other lipid-soluble vitamins like vitamin K and E. And it also secretes the extracellular matrix components like collagens and proteoglycans and fibronectin. And this, unfortunately, when they become overstimulated, can lead to hepatic fibrosis. So this is characteristic of liver cirrhosis, as you get scarring throughout. So notice the scar matrix forming here with these overactivated stellate cells. I've already talked about the liver Asonis concept. So that has to do with the blood flow and the concentration of nutrients. So here we have the central veins, here we have a portal triad, and we can divide the lobule here into region 1, 2, and 3 based on oxygenation and nutrient levels that it receives. Zone 3 is often where toxic injury occurs because the blood flow, nutrient flow, and so on will be much less here. The liver, I said, can regenerate. It remains the same size unless injured throughout life. The cells can actually become activated, and about a little 25% of remaining liver can regenerate into a whole liver again. It's thought really hepatocytes act as unipotent stem cells. There's another type of cell called a valocyte, and this is thought to act as a bipotential stem cell. So it can differentiate into both hepatocytes and cholangiocytes. These are a specialized type of hepatocyte which secretes bile. So they're kind of mixed in with those hepatocytes. As the liver cells regenerate, the functions of the liver are usually maintained. And again, any scarring or cirrhosis can inhibit this. So here's a sequence we'll see with fatty liver is when you get lots of fat in the liver, this can result in fibrosis over time and then liver cirrhosis. So just having fat that's not the only problem. It can lead to the scarring as well with time. And this picture just kind of shows you that whole process and the immune process that goes with it. Embryologically, the liver actually begins from the endoderm. These are the digestive tissues. And I won't go through that whole process here. Basically, you can think of it as a sprouting off from the gut tube. And then the liver develops from that, and so does the gallbladder. So these are from endodermal digestive tissues. In the fetal circulation, just want to review this really quickly. The blood flow from the placenta comes in here through the umbilical vein. This is actually carrying oxygenated blood. It goes into the liver and actually merges here to where the portal vein is coming in. And then we have another duct called the ductus venosus, and both of these streams then converge on the inferior vena cava. Remember, in the fetal heart, the blood is shunted between the right and left atrium in the foramen ovale. So we're basically bypassing the lungs. There's also ductus arteriosus, so blood from the pulmonary artery here is shunted right into the aorta. So both of these bypass the lung circulation. And then the blood comes down the aorta and then leaves via the umbilical arteries. And these umbilical arteries really carrying deoxygenated blood back to the placenta. So notice basically all of this placental blood is going to go through the liver first and then towards the heart. The umbilical vein, when it shrinks and shrivels, becomes the ligamentum teres, and the ductus venosus here is going to become the ligamentum venosum. So those are some of those landmarks I talked about. The liver is the most important organ in blood formation in the embryo, beginning in the 2nd month. So blood cells are made in the liver in the embryo. And the liver of the newborn is about twice the size of an adult liver in terms of relationship to body weight. The liver releases blood stem cells. They migrate to the fetus, and this helps form the T cells. But by the 7th month, the bone marrow essentially takes over blood production. So RBCs are not made in the liver. Blood is not made in the liver after the 7th month of embryonic development. There is sort of a placental-like character, though. You see the connection with the placenta, but we can think of the liver as nourishing the rest of the organism, even in the adult. So again, providing your life juice. When people are devitalized, one of the first organs to think about is the liver. This is really the life-giving inner vegetable system that provides you with all this life activity. Okay, so I'm going to stop the video here, and then we'll pick up the physiology in the next video.